So let us now discuss the fate of pyruvate. Take this as pyruvate, which has 3 carbon atoms in it. Take these as carbon. So this clearly signifies us that pyruvate is a 3-carbon compound. Remember, pyruvate is obtained from the splitting of glucose in the cell cytoplasm. Now, fate of pyruvate totally depends on oxygen availability. And if oxygen is available, it will enter mitochondria. So it will enter mitochondria. Take this as mitochondria. Now visualize pyruvate entering the mitochondria, and it will undergo a process called oxidative decarboxylation, which you can also call it as link reaction. So after oxidative decarboxylation of pyruvate, it undergoes series of chemical reactions such as Krebs cycle and electron transport system, and followed by oxidative phosphorylation, particularly carried out by ATP synthase. And ultimately it can generate abundance of ATP. Now you see, pyruvate has so much potential in them to generate a lot of ATP. So if you want abundance of ATP for your cellular activities, make sure you breathe in oxygen properly. Alright? To sum up, if oxygen is available in the cells, pyruvate enters the mitochondria and proves its potential, that is by generating abundance of ATP. In contrast, if oxygen isn't available, pyruvate will remain in the cell cytoplasm. It will remain in the cell cytoplasm or cytosol, and it will undergo alcoholic fermentation or lactic acid fermentation depending on the metabolic conditions of the cell. So it undergoes lactic acid fermentation as in vertebrates. Example would be our own muscle cells when it undergo lack of oxygen but requires high energy demand. And it undergoes alcoholic fermentation such as in yeast, plants, and bacteria. Now you try to analyze why pyruvate has to undergo fermentation. You know that everything happens for a reason, right? So why pyruvate has to undergo fermentation, especially in absence of oxygen? See, the reason is because to sustain glycolysis. Remember, glycolysis generates 2 ATP, right? So fermentation process will sustain glycolysis to keep producing 2 ATP per glucose. Many people assume that fermentation produces ATP, but it's not. Fermentation is actually, it is a pathway that helps in regenerating NAD+. And by regenerating NAD+, it helps in sustaining glycolysis. Remember, if you recall, glycolysis is a breakdown of glucose into pyruvate. 2 molecules of pyruvate is generated per glucose. So in order to allow glycolysis to keep producing ATP and sustain cellular activities in absence of oxygen, fermentation process operates and helps in the regeneration of NAD+, which gets fed back to glycolysis and sustains the glycolysis. Glycolysis can keep producing ATP in absence of oxygen, and that's how cellular activities get sustained in absence of oxygen. Now, why NAD+ regeneration is so necessary? The reason is because NAD+ is a coenzyme that accepts electrons and gets reduced to NADH during the oxidation of glucose to pyruvate, especially when, if I specify the step, when G3P gets converted to 1,3-bis PGA, there is a consumption of NAD+ in glycolysis, which generates NADH, and NADH is a high energy carrier molecules. It carries high energy electrons, which are used in generation of ATP. So therefore, remember that if NAD plus is not regenerated, glycolysis cannot proceed and ATP production ceases, leading to the lack of energy for the cellular process and ultimately resulting in the death of the cells. So my dear students, be grateful that pyruvate is undergoing lactic acid fermentation in absence of oxygen in our muscle cells, and it is helping glycolysis to keep generating ATP, providing energy for our cellular processes, even in anaerobic conditions.